Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.
Over the last decade, a number of us were thinking that it is time to start to change the way we teach some of our introductory physics courses. And in 2006, we had a chance to begin doing this. This fall, which is now 5 years later than that, the transition is complete. And what I want to do is to share with you something about what we've done and why. Don't worry, I'm not going to cram an entire year of introductory physics into this 1 hour, and there won't be a test at the end. But what I want to do instead is to help you see something of the motivation for what we've done and to give you a flavor of how this new course might differ from the introductory physics course that perhaps many of you took when you were in school. All right. So let's start with a little bit of historical background. We'll go right back to the beginning. This is Aristotle. And human knowledge has obviously come a long way since Aristotle when he wrote this Physica Auscultationis. I'm not really a Latin speaker. The title simply meant lectures on nature. And indeed, physics was everything in science for most of human history. It was about anything there was to talk about. As late as the early 18th century, it still carried this kind of universal meaning. And Harris wrote in 1704 that physics, or natural philosophy, is the speculative knowledge of all natural bodies. Indeed, the only other thing there was at the time was metaphysics, those non-natural bodies. And in that same quotation, Harris says that Mr. Locke, that's the famous Mr. Locke, thinks that God, angels, spirits, etc., which are usually accounted as the subject of metaphysics, should also come into this science of physics. So it was really a universal science at that time. Since that time, when Newtonian mechanics was new, things have kind of changed. Science has had a remarkable run, and it has expanded in breadth and depth in an extraordinary way. We've added to things like physics and mathematics, we've added subjects like geology and chemistry and biology. Biology, after all, a word only invented in 1804, right? Only used for the first time at that time. So we somehow see science now as a much more diverse and separated kind of thing. Fields from foundational ones like physics and math, all the way through the life sciences and into the human sciences. If you look around the College of Literature and Science and the Arts, you will find that most of the departments that are around make some claim to use the kind of approaches and methods of science in trying to understand what they do. So this has been a kind of explosive diversity, and it's a great thing. It's a sign of success, obviously. But it's also kind of a source of risk. It's undeniable that since the time of Galileo, we've learned an enormous amount. It's extraordinary what we've been able to do, and progress has been made on just about every front. I think it's fair to say that we have a pretty firm grasp on the fundamental principles that underlie all of ordinary life. That's an amazing achievement to make in just a few hundred years. Mysteries remain, of course, at both ends of the spectrum. At the most fundamental level, we have questions about things like, why is there mass? And at the more complex level, we have questions about why or what will happen to the climate of the Earth. So there's plenty of mystery that remains, but there is an enormous success to talk about here. The breadth of this success, I'm afraid, excludes today polymaths like Thomas Young. Back in 1807, Thomas Young was one of the great physicists of history, and he gave a series of public lectures, actually remarkably like these public lectures, which he wrote up in a volume in 1807 that compiled the whole one-year-long series of lectures. And that series of lectures did things like cover the entire mechanical arts, introduce kinetic energy into physics, describe the accommodation of the eye, discover surface tension, introduce the concepts of stress and strain, establish the wave theory of light, describe the principles of architecture, and summarize the current understanding of animal and vegetable life. Okay, in one series of lectures. The most remarkable thing is they were incredibly unpopular. He lost the job of giving these lectures very quickly. All right? So he wasn't a great public speaker, but clearly a great polymath. In the modern world, this richness of knowledge that we have kind of makes Young's comprehensive grasp of human knowledge certainly beyond my means and perhaps beyond the means of anybody anymore. But this specialization that's been made necessary by knowing so much has also created some problems, I think. It gives a sense, that an impression of boundaries between disciplines that really don't exist. There are no laws of physics or laws of biology. There are only laws of nature, and they govern all of these things and connect them completely. So today we're beginning to kind of break down these boundaries. I think they're reinforced in a lot of ways by bureaucratic things, like the fact that we have departments of biology and departments of physics that kind of fight for resources with one another and want to have separate identities in a certain way. But we're trying to push back against that and try to achieve a kind of sense of the unity of knowledge, which many people have called for a renewed recognition of, including E.O. Wilson in this pretty famous book from a few years ago. Wilson being the great expert on ants and their societies, and a spokesperson for science in general. So we thought it was time to start thinking about a new course. This is a picture of an old course. This is a picture of teaching physics here at the University of Michigan in 1893. And you'll see that it's not that different. Still have a bench down in front, an audience out in front, some lecture demonstrations on there, and all of that. So we started to think that maybe it was time, in particular in the area of the physics that we teach to life scientists, to create a new course. And we set out to do that. Why a new one now? Well, the courses that we teach actually are remarkably the same as they were 100 years ago. They have a very strong engineering orientation at the time when they were created. The primary goal was to teach people how to do things in the world, to create people who could produce the modern world that we live in today. But this engineering orientation leads people to think that physics is only about pulleys and blocks on planes and inanimate things. We really draw a sharp line that gives people the impression of an unbridgeable divide between physics, the inert world, and biology, the living world, right? So we wanted to try and break that down. The other reason for doing this is that those life scientists, as a physicist, I know that those life scientists cannot understand life without understanding physics. So if we don't teach it to them in a way that makes that apparent to them, there's no way they will know. So this new course focuses on quantitative understandings of the workings of life and try to make students understand how everything that happens in life is enabled by the principles of physics and also constrained by them. Life can't do anything that inanimate matter can't. Right? You can't just make up new rules for living things that don't apply to inanimate things. So when we start off the course, we present them with this kind of prospect. We read them this definition from the Oxford English Dictionary. Biology is a division of physical science which deals with organized beings or animals, plants, their morphology, physiology, origin, and distribution. It's a physical science. This is a surprise to most of the students. They've come in thinking there is this really strong divide between the two. And I asked them to consider questions like this. Do they really believe that biology is a physical science? Of course, we don't require anyone to believe these things, but we want them to seriously take the prospect that life could exist within the bounds of physical laws and come about solely as a result of them. Right? Think seriously about whether that could really be true. And to test that proposal, through the course of a year of physics, they learn a number of physical laws, come to understand these laws of nature, examine how they constrain life, and also how they enable life, how they let life do everything that it does, and ask themselves whether life ever exceeds these laws, whether it ever evades these laws. All right? So that's the kind of overarching principle of what this course is about. And what I want to do this morning is to talk to you a little bit about some examples of things that are drawn from that. So you'll get a sense or a flavor of what it's like to take this Physics for the Life Sciences course. Let's start with a simple example. This is a picture of the Law Quad a few months from now in the depths of January. You all know what it looks like. You go out there, there is no life happening. It's shut down, right? If you come back in June, what you will see is some kind of luxurious vitality, right? Life going crazy, the weeds are growing up, it's just happening. So what's the difference between these two things? What's the one parameter that's changed to allow this to happen? It's just temperature, right? You turn up the temperature and life appears, right? That's the one thing that really has to be there. And this kind of principle really underlies how important that kind of thing is. If we make one physical change to your surroundings, we lower the temperature to this January level, you won't be alive anymore, right? So even something as simple as that is a way to introduce this kind of notion. There is something that provides a really crucial connection and that you might be surprised we talk about a lot in this class. Not the principles or the details of how evolution happens, but that evolution has happened is crucial to this course. Because what happens here is that the laws of physics, the laws of nature, constrain what's possible, right? They put boundaries around what you can do. And what evolution has done is to allow life, through selection of random variation, to explore that range of the possible. Right? Not everything is possible. Some things are impossible. You can only go so far with this. But it allows life to kind of fill those boundaries of what's possible. And the variety of the possible delineates what's there in the diversity of life. So one of the things that I think we can get out of this class that students appreciate about it the most is it helps them to understand why life has the range of diversity that it does, and not more. Right? There are boundaries around it, places where it has to stop. And we talk quite a bit about those boundaries. These questions that are up here now give you some kind of example of that. So many students would come to this class knowing, for example, that hummingbirds eat their own weight in food every day, but not really understanding why that might be. Knowing that the inside of your lungs is an incredibly complex structure, but not really understanding why that has to be. I mean, there are a lot of drawbacks to having such complicated structures. And recognizing that life underwater looks almost completely different from life on land, but not really knowing why that is. Right? So with this course, we can help them to understand some of where this comes from, because much of it comes from the boundaries that are placed on life by physical laws. All right, we're going to start out by setting some boundaries about this. We're going to talk first about a very simple thing, about scaling and size. People often imagine that the world is scale invariant, and this imagination plays out in fiction and in film. So in film, you can see movies like The Attack of the 50-Foot Woman, right? Or The Incredible Shrinking Man, or The Incredible Journey, where scientists are shrunk down and injected into a politician so that they can go in and save his life, right? It's easy to imagine a world which is scale invariant like this. We could make you any size that you want to be. But the world isn't actually scale invariant. It does matter how big things are. And Galileo was the first to really grasp just how much this size mattered. So let's talk about that a little bit. In order to talk about it, we have to think about how a physicist would model an organism. You know, part of the problem with biology is that any given living thing is a very complex thing that is tied up in a history and an environment that's around it and all of that. Physicists don't like that kind of circumstance. It's too complicated. So what we do instead is we imagine everything's a sphere. Right? And we do this because a sphere is simple. A sphere is described by one number, a radius. And once you know its radius, then you know everything else about it. You know its volume, and you know its surface area. This thing, this kind of assumption that an organism is a sphere, very useful one. We'll talk about it a couple times today. It's called the spherical cow approximation, because we imagine that a cow is a sphere and use it. Okay, very simple principle. What does it let us do? It has many virtues, this model. It's very simple, which is a great thing. It means you can work with it. It gives crude estimates for the volume and surface area of something, and more important, a very good idea of how they change with size. Right, about the scaling. Even though they might not get the exact number right for a cow, it would tell you how a cow would change with size. What's bad about it, of course, is it's not very precise. If you want a more precise measure of the surface area of a cow, you need a better model than this. But if you want a rough one, this is the place to start. So let's see where it leads us. First of all, that kind of scaling rule is really shape independent. And we've built some blocks to show you this. So here's a shape, sort of closer to a real cow than our sphere. And if we were to make one that's twice as large, what we would discover is that that thing is twice as long, and twice as high, and twice as thick. Consequently, its surface area, instead of being twice as big, is 4 times as big, and its volume is 8 times as big. So this is quite a bit heavier than this. All right? If we go to a 3 times structure, like this one, okay? It's only 3 times bigger in a certain sense, 3 times taller. But it's also 3 times longer and 3 times thicker. Consequently, its surface area is 9 times bigger, and its mass is 27 times bigger. right? So things, when they scale, they change their surface area and volume in a way that is different from the way they change their length. And if you just write down some things, you can see the details of this model are a little bit different. It's not 4/3 pi r cubed that determines the size of this or the volume of this particular thing. It's more like 1.1 times its height cubed and the surface area of the base. So the details differ. But the basic principle that it varies like size squared for surface area and size cubed for volume, exactly the same. All right? So what can we do with a model like that? Very simple thing, nothing very advanced. It predicts that volume will scale like size cubed and surface area like size squared. So we could put these two together and say, how should the surface area and volume of an organism vary as it grows? All right? And this predicts that the surface area should vary like the volume to the 2/3 power. And since the mass of an organism is proportional to its volume, we can make this very simple prediction that surface area should vary like mass to the 2/3 power. And real observations of animals that grow isomorphically, just meaning they don't change shape when they change size. They stay the same shape, they only change size, show that the surface area does vary with mass in just about this way. And here's one example drawn from a real organism that changes size a lot without changing shape. It's a little amphibian called the dwarf siren. It goes from about 1 gram in mass and a surface area which is just a few centimeters squared, up to 1 kilogram in mass, and follows this predicted law very closely. So very simple principles, very simple thinking about physics can tell you things about organisms. All right? Now that's not that important a thing to know. How does the surface area vary with mass? We might like to ask, what are the consequences of these kind of scaling laws? So let's talk about some consequences. The first one is that the weight of an organism varies like their size cubed. Meanwhile, the area that's available to support that organism, the area on the bottom of this block, varies like size squared. Okay? So the difficulty of supporting that weight, the amount of weight which has to be supported divided by the area available to support it, varies like size. So for this thing, there's a certain pressure underneath it, weight per unit area. Here, for something that's twice as big, that pressure is twice as big. Here, it's 3 times as big. All right? So the actual pressure underneath these things is increasing as they increase with size, just from this simple scaling principle. This means that big organisms will have much more trouble supporting their own weight than small organisms. And you can see this play out very clearly in nature. All right? So as organisms become larger, their legs must become relatively thicker. Not just thicker, but relatively thicker. They have to get thicker faster than the organism gets big in order to account for this. And so we're familiar with this. We're used to seeing elephants with big chunky legs and gazelles with, well, gazelle-like legs, right? It's something that's been familiar. So Galileo wrote about this scaling law right there in his 2 new sciences. On the other extreme, if you keep going down to smaller and smaller size, the problem of supporting yourself goes away almost entirely. And you can choose any shape that you like. And in fact, life has done that. So we see things like crane flies with ridiculous shapes. Absurd, right? And the wading birds have these wonderful, tiny, thin little legs. You just can't make a bird like this that's the size of an elephant. All right? And we have a little demonstration to really try and drive that principle home in your mind. This is one we just made up this week, so I'm not sure how it's going to go. We'll give it a try. All right? So Warren made for me some stilts that are sort of large enough to hold up a person. At least I hope they are. We're going to find out. I'll clamber up here and see. So, nice big legs. I can put my weight on this. You can see that works, right? It's okay. This is big enough for me to put my weight on. What happens if I change it around a little, though? Instead of having a nice big thick leg like this, I have one like the crane fly. Like that. All right? So we can put that in the leg instead. And you already pretty well know what's going to happen here. Right? I can't even really get my leg on it before it breaks, because you just don't scale that way. So you can't make something like that crane fly as big as me, because the materials it's made out of just don't work. Right? They can't support that kind of load. And I'm not going to go any farther with this demo. I think that's enough. hopefully a little visual reminder of how these scaling laws work. Okay. This is important, of course, because it protects us from our nightmares. This is a real gorilla, which is pretty frightening. You know, if you've ever been close to a real gorilla, that's a scary animal. And it's obviously mechanically okay. These things get along in the world. This is King Kong. King Kong is terrifying, right? One of our nightmares. But it's not mechanically okay. All right? If you take this gorilla and you scale it up 5 times, when King Kong stands up and pounds the ground, his arms would break. Right? The materials he's made of aren't strong enough to be scaled up that way. And that's why there is no King Kong, because there can't be. Right? If there could be, there probably would be. All right? So size matters. You can't break the rules. And that's the kind of point we're trying to make. There are laws of nature. They're very, this is a very simple one, and you can't break them. Let's talk about another consequence of size, thermoregulation. So controlling your temperature. Volume determines how much heat an organism produces, because every cell is undergoing processes that generate heat. So the rate at which you generate heat basically scales with your size cubed, with your volume or your mass. Meanwhile, the surface area of an organism determines the heat loss rate, how quickly you lose heat. So the heat made divided by the heat lost scales like the volume over the surface area. So it also scales like the size. So what does that mean? It means if you're small, the amount of heat you're making compared to the heat you're losing is small. And if that's the case, you have to eat like crazy to generate enough heat just to keep yourself warm. And so any small thermoregulating organism, like a hummingbird or a shrew, will have to eat like crazy just to keep up. All right? They're not eating like that because they need to grow fast or anything. They're just trying to stay warm. All right? Lots of these organisms also have adaptations to do things like give up on staying warm and actually allow their temperature to go way down when it's really hard to stay warm, because they're really at the boundary of what's possible. Being a thermoregulating organism that's small like this is really challenging. At the other extreme, if you're really large, then the amount of heat you're making compared to the heat you lose is a big problem. It's really large, right? You're making too much heat. And so losing heat becomes a big issue. And such large organisms tend to be hairless. They have all kinds of mechanisms for trying to lose heat faster than they would otherwise. Okay? So another example of how scaling plays out. There are a lot of details about this, too, that are really interesting to look at. One of the things about life, here on Earth at least, is that life, all life that we know of, lives in either air or water. It lives submerged in a fluid. And those two fluids are really different from one another in a number of ways. And those differences create important differences for life in the air and in the water. So let's talk about just a couple of ways in which air and water are really physically different. The first is density, of course. Water has a density of about 1,000 kilograms per meter cubed. It's dense, heavy stuff, as you know when you carry the milk carton into your house. Air has a very low density, about 1 kilogram per meter cubed. This has a number of consequences, but one of them is that the buoyant forces experienced by things submerged in water are 1,000 times larger than the buoyant forces experienced by things submerged in air. So each of you is submerged in air right now. You are displacing about 0.5 meter cubed of air, and that creates a buoyant force which is about 0.5 kilogram. Very tiny, you hardly notice it. If we submerge you in water, though, you would experience a buoyant force that's about equal to your weight and would almost fully support you. Another difference between air and water is their thermal properties. Water has both a much higher heat capacity. It takes a lot of heat to change the temperature of water. It's about 3,000 times higher than air by volume. And it also has a higher thermal conductivity. So if you're in water, any heat that's in you is going to move into it much more freely than it does in air. And you probably know this if you swim in the Great Lakes, which are almost always very cold. You know how rapidly you cool off when you're in the water compared to what you do in the air. So these differences in air and water provide major pressures on life, and those pressures have played out through something that's usually called convergent evolution. Because the problems are so severe, they get solved by life over and over in basically the same ways, the only ways that they can be solved. So let's talk first about standing up to gravity, whether you live in air or you live in water. So if you live in air, standing up to gravity is a big problem because the buoyancy of the air won't hold you up. Right? It's not enough to keep you up. So you have to be strong. You have to have a structure that can resist the downward pull of gravity. And everything from the largest trees, giraffes, the smallest spiders, everything that lives in air has to do this, has to have a mechanically sound structure that will support it against the downward pull of gravity. This has huge implications for life. I mean, the reason there are so many legs in the animal world and everything else, because you have to do this. You can't get around it. It's one of the governing principles. In water, on the other hand, buoyancy makes gravity really irrelevant. And particularly for organisms that live entirely in the pelagic zone, not at the surface, not at the bottom, but in the middle of the ocean, you know, they just don't have any gravity to deal with. And so the kinds of organisms that you see take on whatever shape they want. You know, this is a siphonophore, a kind of cnidarian that, related to the jellyfish, that just is like a piece of tissue that's 30 meters long. You know, and it can be that way because it lives in an environment where gravity is irrelevant. You can't be that way in the air. Another famous example of convergent evolution has to do with flight. If you want to stay up in the air, buoyancy is not going to do it for you. So if you want to stay up against the downward pull of gravity, you have to push down on something so that it will push up on you and resist gravity, right? So the way to push down on air is to have something large enough to be able to push a lot of air downward so that you generate a large enough force. And of course, this solution to this problem has been repeatedly evolved in the history of life. Insects did it first, then reptiles did it. Well, birds are probably related to the reptiles, although not these reptiles. So it's an independent evolution of it with birds. And finally, mammals have done it. Repeatedly, over and over again, evolution comes to the same solution because there's really no way around it. And all of these do the other thing that helps with this, which is to reduce their weight as much as possible with things like hollow bones and very thin wing structures. Okay? If you want to move through air and water, the differences are really different. I mean, the properties of air and water make that really different. Moving through the air is really pretty easy. And so it doesn't drive much change in organisms. You can take on any shape or size that you want and still be able to freely move through the air. So we see things with kind of ridiculously blunt-faced structures because it's okay to be that way when you're moving through air. Air can't stop most organisms that live in it, and it certainly can't stop this guy. Okay? Water is completely different. When you go to move through water, the friction you experience is, roughly speaking, 1,000 times larger than when you move through air. So if you've ever taken one of these classes where you try to run through the water, it's actually really tough to do. Even Dennard would find this difficult. Okay? So what has happened? Evolution has repeatedly come to the solution that you must be as streamlined as possible. You have to work to reduce that friction. And so you see it in all organisms that try to move with any speed through the water. And that can be the sharks and the fish, marine mammals, marine invertebrates like this, reptiles, birds, everything that wants to work in the water has to deal with this problem. And so it has to come to the same kind of solutions. There's really no choice. All right? Staying warm, another big problem. In the air, it's actually not that hard to stay warm. As we saw, the heat capacity of air is not very large. Its conductivity is not very large. So it's relatively easy to stay warm in air. That's why there are many warm-blooded organisms that live in air. Staying warm in water is a much bigger challenge. And you find that there aren't many warm-blooded organisms that live in water. The ones that do take advantage of a variety of tricks. First of all, they insulate. And they may insulate either by using fat, as many marine mammals do, or by using air, as most of the marine birds do. They trap air in their feathers, and that air that's trapped in their feathers is a very good insulator. In those cases where they have to have contact between their body and the outside world, they use an amazing mechanism called countercurrent heat exchange. Let me spend just one moment to explain this. If you're a duck, or a merganser in this case, and you stick your feet down in the cold water of the Huron River, it's cold, right? You don't want to lose all that heat to your feet. So what you do is you take the blood that's hot, that's coming out of your body and going down into your feet, and you run it right next to the blood that's cold and coming back up into your body. So as soon as that hot blood gets near the cold blood, the heat leaves it and goes into the cold blood and comes back into your body, instead of sending the heat down into the water and losing it to the Huron River, right? So if you evolve a network of veins and arteries that send blood out this way in close contact with the blood that's coming back, not draining from one to the other, but right next to it so that the heat can freely flow between the two, then the heat will never leave. It's called countercurrent heat exchange. There's a picture of it here. There's a flow of cold blood coming into the body here and a flow of hot blood coming out, and the heat gets exchanged across like this so that the cold blood coming in starts out cold, and by the time it gets to here, it's hot again, and the heat is flowed through here instead of down into the river. That happens not only, it has again been repeatedly evolved, it happens not only in water birds like this, but in many, many other organisms that are trying to maintain a temperature difference between different parts of their body. The tuna does this with a truly fantastic thing called the rete mirabile, the miraculous network inside its body, where there's so many veins going intertwined like this that it makes the flesh look completely different. You know that central part of a tuna steak that's really red? That's what that is. And the tuna does this so that it can maintain an elevated body temperature in cold water. It's one of the premier hunters in the fish world. It can swim at 50, 60 miles an hour. It does that because it keeps its temperature up. Right? If it was cold-blooded, it couldn't do that. Swordfish do something similar, but even weirder. They actually fish quite deep often and need to be able to see. It's really dark down there. It's also really cold. So what they do is they have a little network like this that allows them to heat their eyes. Not their whole bodies, just their eyes. So their eyes function better when they're deep underwater. All right. Now, when you breathe water, it's nearly impossible to maintain an elevated body temperature. And that's because in order to extract oxygen from the water, you have to put your blood in very intimate contact with the water. And if you do that, then the heat flows out of it very freely. So if you're going to breathe water, as most fish do and so on, it's really hard to maintain an elevated body temperature. So very few of them do. Almost all are just cold-blooded. They give up on this problem and decide to adapt their biology to live at the temperature of the water that surrounds them. So when you think about warm-blooded things that live in water, you know, they're mostly these things that breathe air because they avoid that problem. Okay. That's all about organisms on the large scale, things that are sort of like machines. They're kind of familiar. Their problems are kind of familiar to us. But one of the things that makes life so remarkable is that it operates on this tiny scale inside this cell on the smallest scales. And understanding the sort of vitality, the thing that makes life seem completely different from inanimate matter, requires that we should begin to understand how organisms work on these tiny, tiny scales. And of course, the advances in biology for the last 50 years have come in large part due to the ability to begin to understand life on this tiny scale. The field of molecular biology, a clearly interdisciplinary field, was born of advances in physics and chemistry. And I'm going to talk about two different things here to give a little flavor of some of the things in this realm that we might teach in this course. So I'm going to talk first a little bit about statistical physics, about how the atomic picture of matter gives us an understanding of one of the main mechanisms for cellular function. And then I want to talk about X-ray diffraction and how it has allowed us to open the world of structure determination. Probably you've read in different places that proteins are really important for life, also things like DNA, and that their structure determines their function. So to begin to understand how any of that works, we had to learn how to determine the structures of these molecules. What I propose to do before we're done is to teach you how X-ray diffraction was used to determine the structure of DNA. Okay, so those are the two things we're going to do for the remainder of our time. First of all, let's talk about just basic statistical principles. If you want to move necessary materials from place to place on a large scale, it's easy. You take a gulp of air, right? You move air into your lungs, no problem. You take a swallow of food, whatever it is. But this doesn't work on the molecular scale. If you want to get one molecule of oxygen and move it over there are no tweezers you can use, you know? You can't buy a little truck that will carry it from one place to another. And so what life does to move materials on the molecular scale is to use purely random motion. It doesn't move them. It lets them move themselves, all right? So molecules, atoms, at any temperature other than absolute zero are always in motion. And life takes advantage of this motion in order to achieve motion, achieve the movement of things on molecular scales that life really needs to achieve. Okay, let's talk a little bit about how that works. Random motion, most of the time, leads to fully balanced flow from one place to another. If it's just random, how could it be going in any given direction, right? So the kind of balanced flow that it gives rise to is illustrated in this picture. So here's a picture where we have lots of oxygen on one side and lots of oxygen on the other side. Now, because of random motion, these atoms are going to move around. Some of them are going to move from the left-hand side to the right side. Some of them are going to move from the right-hand side to the left side. That will generate a flow. Just due to random motion, there'll be a flow to the right and a flow to the left. If these two things are equal, those flows will be equal. Right? There'll be no net flow because it's random and it's equal on both sides. But now let's just change that a little bit and put a lot of oxygen on one side and very little oxygen on the other. The same random process, no guidance, no force involved with it at all, just random motion, gives rise to a large flow to the right and a much smaller flow to the left. It's just random chance, nothing more than that. No force involved with it at all. But it does create a net flow. So if a circumstance can be created where there's not much oxygen on one side and there's a lot of oxygen on the other, you don't have to move the oxygen. It will move itself. All right? And so by setting up circumstances like that, life enables oxygen to flow where it needs it to do so. All right? So life uses this transport in a number of ways. I'll tell you about just two of them in a very basic way. To deliver molecules across short distances. And one of them is moving oxygen. So when you breathe, oxygen is brought very close to a permeable boundary, a boundary that it can pass through. This happens inside your lungs. Remember that very complex structure in your lungs we talked about? Your lungs are actually broken down into an enormous number of very tiny alveoli into which air is pulled. All right? So you let the air go into this alveolus, and right next to it is a capillary that has blood in it. There is a low concentration of oxygen in the capillary and a higher concentration in this alveolus. And so, just because of random motion, it moves across that boundary and into the capillary. This is the reason why you have to have such a complex structure in your lungs, because diffusion does not allow oxygen to go very far. So you have to get it very, very close together in order to allow that to happen. And so the way your body does that is to make many, many tiny things inside your lungs, each of which you can get really close to the oxygen in. So although there's a lot of cost associated with that, it makes your lungs a very fragile and complex structure. It's essential that it should do it. And so that's what happens inside your lungs. Fish do the same sort of thing. They have gills to do this that allow them to have an enormous amount of surface area so that that oxygen can go across it. Another place where diffusion plays a really important role is in nerve cells. Nerve cells become electrically charged and then can be discharged in order to transmit a signal. They become electrically charged through diffusion across a membrane that can change its permeability. So the secret in nerve cells is that they have pores that can open and close to allow ions to go across in one state and then close and not allow them to go across in another state. So they can open up, let some ions flow across, then they can close and not let ions flow across. And that allows them to constantly change their charge state. All right? Same random process being captured, used by life in order to move ions on these very small scales. So diffusion, one of the central processes, could only be understood by understanding an atomic picture of matter, really. What are the limits for these things? Well, the flow rate depends on a number of things. It depends on the concentration on both sides, the temperature, the permeability of the membrane, how much surface area there is to go through. If there's only a tiny little spot, you don't get much through, right? And if we remember the basic scaling of these things, you can recognize that the oxygen required goes like size cubed, and the oxygen delivered goes like size squared, like surface area. So the required oxygen over delivered oxygen is proportional to size, like the other things that we've talked about. And that means that delivering oxygen by random transport gets harder and harder as you become larger. It's really easy if you're a single cell, because your surface area to volume ratio is really large. So you don't need any lungs. You don't need anything complicated. Whatever is outside you that is not inside will flow in freely due to diffusion. All right? But as you get larger, this ratio of volume to surface area gets bigger and bigger. And so in order to get around this problem, organisms change shape and not just size. So what you do is you add many folds and branches in your lungs, and fish do that with gills. Insects don't do this. Some of them do have things called tracheae, but most of them take in oxygen directly through their surfaces. And this kind of evolutionary trap, if you like, is one of the reasons insects have never gotten larger than they are right now, because they run up against this kind of boundary. Okay, that's one topic in the microscopic structure of life. Let's go and talk about a big one that everyone's familiar with. How is it that life shares the instructions for how to construct a cell? So this was a really central problem at the beginning of the 20th century. It was known that there were laws of inheritance that were very complex and could be studied in great detail, but the mechanisms of it were completely mysterious. And beginning in around the 1940s, physicists also started to really think about this. And in a really seminal work, Erwin Schrödinger, who you all have heard of, at least because of the cat that he talked about, if nothing else, wrote some essays during World War II when he was staying in Dublin about life, about the physical aspects of a living cell. It's a fascinating thing to read because it gives you a clear idea of just how vague the understanding of life was at the time. He had a real sense, a good guesses about how some mechanisms of life might work, but really it was a great mystery at the time. So in the beginning there was a lot of speculation. Eventually there was an identification of what the mechanisms were for transmitting inheritance in life, and ultimately the details of the structure of the molecules that do that were determined. I want to talk a little bit about something done a little bit after Erwin Schrödinger, a paper written by Richard Crane, one of the great physicists here at the University of Michigan, in 1950, called Principles and Problems of Biological Growth. Okay? So Dick Crane wrote this paper at a time when this was purely speculative. And he said, all right, let's not try to figure out exactly what the molecule is or anything. Let's just think about simple principles. I know we want to have a molecule that can produce itself, that can self-assemble, right? And if we're going to have a structure like that, it must have its pieces connect together always in the same way. So a very simple idea that if you have two different kinds of pieces that go into that molecule, at least there must be only one way for them to go together. So maybe the way is this one goes here and then this one goes here, right? And then if you put another piece on, there'll be one like this and one like this, right? Just a simple rule. No understanding of exactly what it is. What he discovered was that if you apply only that rule, you come to a conclusion. Every structure that is built according to the one rule laid down will be repetitive along a screw axis, which is to say that it will be repetitive as seen by an observer who rotates around the axis as he moves upward or along it, whether or not it's repetitive seen by an observer who does not rotate. If the structure is broken into pieces which are equal in repetition length, all the pieces will be identical, and so on. He was an experimental physicist, so he didn't just figure this out in his head. He got little things and made repeating structures out of them. These are pictures from the article. Matchboxes. Right? Nice repeating structures. So he made a rule. This red top matchbox attaches to the next one like that. And then he attached the next one in the same rule, and the next one in the same rule. Oh, and he got a helix like this, right? Then he tried another rule of connection. Here's a different rule of connection between matchboxes. Doesn't matter what rule of connection you use, you get a helical structure like this. Okay? This is in 1950, 3 years before the structure of DNA was discovered. Okay? He made other models just to prove it to himself. Could I come up with any rule that wouldn't give me a helix? He made this one out of bobbers and corks. All right? Pretty fantastic that a simple principle like that could give you an idea that would turn out to be so relevant for understanding replicability in life. So, one of the great achievements that he made. Let's talk about DNA structure. So, the story of the uncovering of DNA structure is one of the great dramas in 20th century science. It involved a group of people who made measurements of X-ray diffraction patterns, especially Rosalind Franklin. It involved people who were really chemists, like Linus Pauling, worked on mathematical physics and chemistry, and it involved these two famous characters, Watson and Crick, who in 1953 published a structure for the DNA molecule that we now know was correct. I've labeled these people according to the fields that they were trained in, to make a point, right? This is an interdisciplinary science. We have physicists, we have chemists, we have a zoologist thrown in, right? People came to this with different views, and that was essential to being able to make the kinds of advances that have been made in the life sciences in the latter half of the 20th century. So DNA structure was determined. The key clues came from a single picture. And this is a copy of it, right? This is the picture from which Watson and Crick were able to infer that DNA is a double helix with certain particular properties, okay? Let's blow it up a little because we're going to talk about how they were able to use this picture to figure out that DNA was a double helix. By the time we're done, in 13 minutes, I want you to understand that, okay? All right, I think we can do this. There are 4 clues. The first one is called the layer lines, and let me label them for you. Here they are. See these lines that go straight across? Those are called the layer lines. That's the first clue. We're going to explain where they come from. The second is the central cross. Hard to miss that in the picture. The third is the missing fourth layer line. Here's the center, first, second, third layer line. Look at this. The fourth one is missing. Another important clue, crucial clue actually. And the last bit are these big bright dots at the top and bottom. Okay? Those four things are the clues that were necessary for Watson and Crick to figure out that DNA is a double helix. Let's see where they come from. You need to know a little bit about how light diffracts in order to understand this. So we're going to do a demonstration and show you the pieces that you need to know about. Very simple things. So there's a laser up on the wall there right now. It makes a dot on the wall. What I'm going to do is I'm going to put in front of that laser a single slit. And if you see that single slit in front of the laser, what you will see is that it spreads the light out sideways. Okay? So if I put up a single slit like that, the light will spread out perpendicular to it. That's fact number one that you need to know. What do you think would happen if I took my slit and I turned it sideways? The light would still spread out perpendicular to it, but in a new direction, right? Remember that, because that's going to be important. If I put together light from two or more slits, then the light from each slit can cancel the light from the other slit. And we can end up with separated spots, kind of like what you see here. And let me show you that. I'm going to put light through a set of a couple of slits here now. And you see how there's now dark spots in what used to be just a solid band of light in the middle. Used to be like this, a solid band of light. Now it has dark spots in it. That interference between them will tell us that there's more than one thing. And not only that, it will tell us how those things are spaced. If those two slits are very close together, then the dark spots will be the opposite. They'll be very far apart. And if those two slits are very far apart, then the dark spots will be the opposite. They'll be very close together. Okay? Three principles. Here's the other one. These three principles going together say that light going through a slit will spread out perpendicular to it. If it goes through multiple slits, it will leave spots, dark spots like that. The light will appear in only separated spots. And if you have slits that are close together, they will produce spots that are far apart. And if you have slits that are far apart, they will produce spots that are close together. Three principles. That's what you need to know. OK, let's go and explain these phenomena of DNA. Here's basically the way the experiment works. X-rays, just an energetic form of light, are impingent on DNA, the nice double helix that you're familiar with, and run into it in this way. Okay? They run into it in this way. And what we're going to see is that there are a number of things that matter here. There are three spacings in this double helix molecule. One of them is the whole pitch of the molecule. It starts out here, it goes around one cycle, and comes back to there. That large distance we call p. Okay? All you have to remember is that there's a large distance. Then there are two smaller distances. One is the distance between each of the steps on this ladder, which is 10 times smaller than the big distance. It's 1/10 as big. So we have a small distance and a large distance. Then there's an intermediate distance, because DNA is not just a helix. It's a double helix. There are two helices that are separated from one another, not by half of this distance p, but by 3/8 of this distance p. Okay? So those three spacings will all be important. There's also an angle in this, what's called the pitch angle. If you took DNA and you stretched it out more, then it would be more like a straight line. And if you squashed it, it would be more like this, with steeper steps, right? So that angle will also come into this. All right? So let's go through and explain the four things that we talked about. Here's the first. There's a large distance in this molecule, the p. A large distance gives rise to dots that are what? That are close together. Excellent. So we should see a close spacing in that pattern. That close spacing is the layer lines. Okay? So I'm going to show you this using a little slide that has nothing but lines that are spaced like this on it. Okay? So we're looking at just the first piece of the DNA structure. And if we do that, what you see are, over here on the wall now, are a bunch of dots spaced close together. A bunch of lines like that produce a bunch of dots spaced close together. First thing in the pattern, layer lines. So there must be some large repetition pattern in the molecule because you have closely spaced layer lines. That's the first thing they learned. Okay? The second thing is the cross. So what creates the cross? Here we have these long lines, and they send light out perpendicular to them. Remember, a long slit sends light out perpendicular to it. So what would happen if we took those long slits and we tilted them on the side? They would now send light out perpendicular to them. Instead of straight up and down, they would go out perpendicular to them. So let me show you what happens if we take the same set of lines and we tilt them in one direction. Okay, if you look over there now, we've tilted them in one direction. Okay? All you're looking at is a set of lines that are tilted over in one direction, and they produce that one-sided pattern. If I tilt them the other way, what do you think is going to happen? So we tilt them the other way, they go the other way. All right? Now, DNA is not tilted in just one direction or the other. Because it's a helix, it's actually tilted both ways. Right? So it has a set of things tilted this way, which produce a pattern going that way, and a set of things tilted this way, which produce a pattern going that way. Okay? So now we've made the X in the middle. The layer lines are still there because the spacing between these things is still that large P. So we still get close layer lines, but they now appear along the X. Okay? So this told Watson and Crick that there was this big repeating spacing, and it must be a helix. Did it tell them it's a double helix? Well, there's no clue in this that it's a double helix yet. So if it was a single helix, it would look like this. In fact, they didn't even have to work hard because Linus Pauling had figured out that this was the signature of a helix a couple years before, and everybody who saw a cross in an X-ray diffraction pattern immediately said, oh, it's a helix. Or you don't even have to be smart. But the next clue is a little more subtle, and I'm going to kind of gloss over it a little bit. But let's show you what it is. So the next thing that we noted in the 4 things we listed, the layer lines, the cross, the missing 4th layer line. The missing 4th layer line. Okay? Where this comes from is it comes from the fact that it's actually a double helix. And it has a spacing of p between each of the helices. So each goes around in that distance p. But the two are offset from one another by 3/8 p. All right? The consequence of that is that light that scatters off this helix interferes with light that scatters off that helix destructively at the 4th layer line. Okay? So you have layer lines 1, 2, and 3. They're actually slightly different intensities, too. That's another clue about this. But what you get is perfect cancellation at the 4th layer line. So if I send light through something that looks just like the picture that you see here, a double helix with no steps on it still, just a double helix. Let me see where that is. That's just the next step down. Then what we'll get, let me get it in there. Let's see if we can see it. Can we see the layer lines over here? Yeah, you can see them. Not great, though. You can see 1, 2, 3, and then you see a missing part. You can see it's darker there, and then it kind of comes back a little bit. Okay? So that missing 4th layer line, that was the clue to the double helix. And of course, the double helix of DNA is pretty crucial, right? Because it lets it unzip and have 2 copies, right? The 2 copy part is crucial because that's what lets you replicate it. So that was the clue that told him it was a double helix and not a single helix. The last point is easy. We save an easy one for last. There are a bunch of things really close together, steps along the DNA pattern. If they're really close together, what kind of spots do they produce? Far apart. Okay, so the long baseline pattern makes the close together layer lines. The close together things make the dots that are far apart. And if you look at this, if you count out, there are 1, 2, 3, missing 4th, 5, 6, 7, 8, 9, 10. It's 10 times closer together. It should be 10 times farther out. And that's where it is. And they appear both on the top and the bottom. Okay? So if I go to the pattern on this slide, which produces a kind of DNA-like pattern, we should see something that looks a lot like what Watson and Crick saw. Let's see. So you can see the big dot on the top, at least. You can see it pretty well up here. That's that dot that's produced by that larger pattern. Okay? There it is. Now you know, when you go home, you'll be able to explain to everyone why this picture in the front of their biology book told Watson and Crick, of course, it's a double helix. It didn't happen quite that fast, but it didn't take a lot longer. It was really pretty quick that they figured out, once they had good data like this, that it was actually a double helix that drives it. Okay? All right, my goal here was to give you some flavor of the kinds of things that we've tried to include in this class so that people would find it difficult to avoid the connection between the sciences, to see a kind of unity in all this. All right? There's no question that beginning to understand the mechanisms of life is one of the great triumphs, really, of 20th century science. And it's crucial to recognize that achieving it required seeing life's essentially physical nature and abandoning vitalist notions that life obeyed principles that were different from inanimate matter. If we hadn't crossed that line and followed that path, we would not be where we are in the life sciences right now. With this course, we've begun to teach physics in a way that recognizes this great success and acknowledges, maybe I hope, more openly the unity of science. Now, while the successes in the life sciences are remarkable, it's clear that there are some really grand challenges that remain, and I want to close with just a couple of them. So if life is purely physical process, then its origin here on Earth should be both understandable and ideally replicable. Right? So the origin of life problem remains a really interesting one for scientists of all kinds to work on. I think there's been kind of a backing away from it for a while. Maybe it's beginning to return now. It's something that people really need to work on. The last decade of astrophysics has made it clear that planets are ubiquitous in the cosmos. And we have also learned that the conditions of life are probably much more widespread than we imagine, inside the moons of outer planets and things like that. So the conditions for life seem to exist all over the cosmos. If life is a purely physical process, then I can think of no reason why it shouldn't be extremely widespread. And we need to show that it is the case. We can't do that right now. Our instruments are just getting close to being able to. It's very important that we should find out whether it does. Closer to home, of course, there's innumerable mysteries about the life that remains here on Earth. Like one that really fascinates me is how is it that the genetic code is turned on and off in such a way that it allows the development of an organism? So that the same sequence of DNA is present in every one of your cells, and yet your cells turn into many thousands of different kinds in this hyper-coordinated way. That's a fascinating, complex, but fascinating problem. And finally, the last one, the most personal question of all, is really a question about what we all are really, each of us, right? Is all this rich sense of self-awareness that you have, this sense of consciousness, the thoughts and feelings that you have, are they really just physical things? At this point, it's very hard to accept that that's true, but we have no real solid other way of understanding them. So that effort to try and understand how the mind and consciousness work is one of the great challenges of all. And I think that the next century will probably be heavily engaged in trying to do that. So we're far from final answers to these questions, but there's one thing about them that I'm completely sure of, and that is that the answers to these questions will not come solely from physics, or from chemistry, or mathematics, or biology, or philosophy. But they will come from bringing together all of these different approaches to human knowledge. And that's what we're hoping that a course like this will help to emphasize to students at this early stage in their careers. Thank you.
Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.